Hi, my name is Yuliet. I am a first-year chemistry PhD student in the Tretyakjova group. Natalia is actually cross-listed in the chemistry department as an adjunct PI, so it was a great opportunity for me to join her group in the medchem department. My research is in mass spec analysis of protein adducts. So when you are in your environment, you get exposed to all these different chemicals, and those chemicals can actually adduct to the hemoglobin in your blood, and we can take that and we can analyze what you might have been exposed to from that on liquid chromatography mass spectrometry. The primary goal of that is to eventually connect that to different ethnicities exposure and then to different cancer risks and to address some disparities there. So that's really important. I greatly appreciate it because it gives me an opportunity to learn these fantastic techniques that are really versatile while at the same time advancing medicine and advancing chemical research. So since joining, I have been placed primarily under a senior student, and I have learned quite a bit about how to take care of the instruments and how the different adducts work and how the different flows go. And then I meet with him and he meets with Natalia, and we both meet with Natalia together to discuss this work and how we're working together to move forward as I transition in and he transitions towards finishing his doctoral work. I really appreciated that sort of leveled mentorship from both of them, and that's been very fantastic for me. I would say that my primary piece of advice to any prospective student is to spend a great deal of time in the lab and with the people that you expect to join, because it is very, very important to make sure that you click with the other people in your group, not just that you like the research. And I found that with Natalia's group, including quite a few different chemistry PhDs and medchem PhDs, you get a really nice diverse group of people, and some really great advice and multiple different perspectives really bring that forward and help things go. My background is that I was a double major in chemistry and forensic science, and forensic science itself is a very interdisciplinary science. You take facts and procedures from many different areas, and I find that when you have multiple people coming together, you can address different problems at different points. So because we have synthesis and cell biology and mass spec, we can address at every juncture what could be done better, what could be possibly going wrong. We have synthetic chemists double-checking our structures of our adducts for the analytical work, which explains why our masses weren't quite right. And then we have our cell biologists confirming that they're using the correct tools to update the incubators from the mass spec people. And it's just really going to be very beneficial to work together as a cohesive unit and to get different ideas and perspectives from different people and different specialties, because all of those specialties together make this research work. You don't just deal with cancer in a clinical setting. You deal with it at every stage backwards, from the clinical to the biological to the cells, and then to the chemistry that actually causes those changes in cells, and then you see that on the macro scale in the clinic. The University of Minnesota really promotes interdisciplinary work, so there's a lot of opportunities to work with other PIs outside of your group or to be co-advised by two different PIs, and I think that's a really unique part of the University of Minnesota. So I think that's really valuable to me, is that you've got a network that goes beyond the lab at the University of Minnesota.